everyone. Thanks so much for joining us today for this month's coffee chat. My name is Bamboo Dong, and I'm a Scientific Affairs Liaison at Menarini Silicon Biosystems. Today we'll be talking about the development and validation of a new assay for the isolation, enumeration, and characterization of circulating melanoma cells using the CellSearch system. The methods we'll be discussing today are intended for research use only and should not be used in diagnostic procedures. As many of you know, melanoma is a cancer of the skin cells that bear pigment. There are roughly 100,000 new cases every year, but one of the biggest challenges is in the early diagnosis and monitoring of this disease. At present, there are no blood biomarkers for melanoma, so current standards of care involve full body imaging, physical exams, and biopsy. Because treatments can change as often as 6 weeks, there's a need for easier, less invasive, and less costly monitoring methods. One possible option may be circulating tumor cells, which can be obtained from a simple blood draw and have proven to be valuable in the prognosis and monitoring of other diseases, such as metastatic breast, colorectal, and prostate cancer. As with carcinomas, the treatment of advanced melanoma is complicated by its heterogeneous histopathology and continuous mutation in the metastatic setting. By using an accurate and reliable method for the enumeration and characterization of circulating melanoma cells, or CMCs as we will refer to them for the rest of the talk, it may help facilitate the development of more effective therapies for metastatic melanoma patients. There have been some conflicting results in previous studies on the prognostic value of CMCs in patients, potentially due to the use of different detection methods, which ultimately really highlights the need for a standardized assay. So how can the CellSearch help? For those unfamiliar with the system, the CellSearch technology was the first and only clinically validated FDA-cleared technology for the enumeration of circulating tumor cells. It's been extensively characterized and used in over 650 peer-reviewed publications in the oncology space. To use the CellSearch system, blood is first collected into cell-safe preservative tubes, which stabilize cells for up to 96 hours at room temperature. After sample prep and centrifugation, cells are loaded onto the CellTracks auto-prep system, which automatically prepares the sample. Target cells are enriched for using ferrofluid nanoparticles and then further stained for easy identification. Once ready, the enriched sample is transferred and further incubated in the Magnus cartridge holder, which uses a magnet to pull the cells onto a single plane where they can be analyzed on the CellTracks analyzer. Let's take a closer look at the CMC assay and the steps behind its development. Step 1 was identifying the appropriate antibodies to use for the capture and identification of melanoma cells. We evaluated several combinations of capture and detection antibodies, and then after selecting the one we found optimal, we evaluated the accuracy, sensitivity, linearity, and reproducibility of the assay by using healthy blood spiked with varying levels of SK-MEL28 cells. After we verified the results of the assay, we evaluated the stability of the SK-MEL28 cells up to 96 hours after being spiked into the blood to test the stability of the antigens. The specificity of the assay was evaluated in healthy volunteers, and the incidence of CMC detection was explored using blood from patients with metastatic melanoma. We also evaluated the prognostic significance of the presence of CMCs in a small set of patients with metastatic melanoma who were followed for overall survival. Lastly, we looked at the feasibility of further characterization of the captured CMCs using a Ki67 protein marker. Here's the final kit and the cell selection markers, which we'll take a closer look at on the next slide. It's worth noting here that for researchers looking to use additional markers of their choice, the CellSearch also has an open FITSI channel, which can be used for a user-defined antibody. For the research use only CMC assay for CellSearch, we use ferrofluids coated with CD146 antibodies to immunomagnetically enrich both melanoma cells and endothelial cells. Nucleated cells are identified using DAPI, and melanoma cells are positively selected for using a PE conjugated antibody that binds to the high molecular weight melanoma associated antigen, or MEL. a mixture of two APC conjugated antibodies, CD34 and CD45, are used to identify and exclude endothelial cells and leukocytes. Here's an example of what someone would see from the CellTracks analyzer. In order for a CMC result to be considered positive, it must be MAL positive, DAPI positive, CD34 negative, and CD45 negative. With the open FITC channel, researchers can apply a user-defined antibody. In this case, we wanted to evaluate the ability of the assay to further characterize isolated CMCs using Ki67, a protein marker specific for proliferation. All the images presented here are classified as melanoma cells. However, not all are positive for Ki67. You can see the cells in the top three right images here show strong staining for Ki67, indicating that they're in a state of proliferation. The second image, with two nuclei, appears to be actively dividing, although you can see from the fourth image here that although this cell also has two distinct nuclei, it's not expressing Ki67. All of these images are from the same patient, so you can see there's heterogeneity in the expression of markers. One thing that's interesting about some of the CMCs that have been captured from patients with metastatic melanoma is that you can see they come in a variety of shapes and sizes, ranging from 4 microns to over 30 microns, with varying nuclear to cytoplasmic ratios. Some of the cells are multinucleated, and we have even observed small clusters of cells in some patients. It's one of the advantages of the CellSearch system, as opposed to some other methods that isolate based on size. If you were to use a size selection system, you'd potentially be missing out on cells, not only for detection, but also for downstream molecular analysis. During the evaluation of assay performance, varying levels of SK-MEL28 cells were spiked into 7.5 mils of blood collected from normal donors into cell-safe tubes. Samples were processed 24 hours after spiking, with testing performed over 2 different days using different instruments and operators. The percent recovery was determined, and the actual number of cells observed in each sample was plotted against target spike levels. Overall, the average percent recovery was 88%, with linear recovery and a Pearson's correlation coefficient and R-squared value of 0.99. One of the unique things about the CellSearch system is the cell-safe blood collection tube, which allows for the stabilization of cells at room temperature. In this case, we looked at the stability of CMCs collected into cell-safe tubes using SK-MEL28 cells spiked into normal blood from healthy donors. Samples were processed 24, 48, 72, and 96 hours after the time of spiking using the CellTracks CMC kit. We found that average recoveries at each spike level were not statistically significantly different at any of the time points compared to 24 hours. Our primary goal in developing this assay was to create a standardized, research-use-only assay for the isolation, enumeration, and characterization of CMCs using the CellSearch system. It's a reproducible assay that can be used on blood samples collected into cell-safe tubes for up to 96 hours. But to further define and validate the potential clinical utility of CMCs, more clinical studies enrolling appropriate numbers of patients will need to be undertaken. I did want to highlight a recent key publication using the CMC kit for CellSearch. This was a paper published last year by the Lucci Lab at MD Anderson. The purpose of the study was to explore reproducible biomarkers for patients with stage 3 melanoma. In their study, they isolated and enumerated circulating melanoma cells in 243 patients with stage 3 melanoma and compared relapse-free survival in patients with at least one baseline CMC versus without. What they found was that patients with one or more CMCs at baseline showed significantly decreased relapse-free survival within 6 months. 23% of the 90 patients who had at least one or more CMCs at baseline relapsed within 6 months, compared to the 8% of 153 patients who did not have CMCs. Furthermore, at both the 6-month and 54-month follow-up, patients with one or more CMCs detected at baseline had lower relapse-free probability than patients with no circulating tumor cells at baseline. If you want to take a closer look at this paper, we will provide a recording of this talk afterwards that you can stream anytime. If you have any questions about how you can incorporate the enumeration of CMCs into your melanoma research, please let me know or reach out via email. We do have global lab services as well, and I'm happy to provide more information regarding